I would like to start with a disclaimer saying that I'm not an expert in allostery at all, so I'm probably the only synthetic chemist in this room. And I really thank Giovanni for having invited me because he thought that I could have something interesting to say to this audience, and I hope I won't disappoint the expectation. So for this occasion, I was undecided between two different topics. On one hand, the use of deuterium in drug discovery and development. On the other, our work on IDO1 protein. And I went for the second topic, as you can guess from the title of my presentation. But let me share with you this slide where you can see a very interesting molecule. This molecule is eucravacitinib and was approved by EMA, at least in Europe, for the treatment of psoriasis last year. This molecule is thought to become a game changer in the treatment of autoimmune disorders. And this is due to two peculiar properties that eucravacitinib has. The first property is that it's an allosteric inhibitor, so that's why I'm talking about this molecule now. And it is very specific for one kinase, TYK2, which belongs to the JAK family. And this specificity is due to the fact that it binds to an allosteric site, which is represented here. But the problem is that this molecule in vivo undergoes an oxidative metabolism, which is an anti-alkylation of the secondary amide, leading to the formation of a non-selective primary amide. So it was a compound, the one with the hydrogen atoms, that was very specific in vitro, but that lost completely the specificity in vivo. And so the second feature that this molecule has is the presence of these three deuterium atoms. So the introduction of these deuterium atoms in this molecule allowed to slow down the oxidative metabolism mediated by cytochrome and to retain specificity, not only in vitro, but also in vivo. And this is due to the so-called deuterium kinetic isotope effect. And this is something that I'm really passionate about. And so this is why I wanted to show this molecule to you today. But the main topic of my talk is given by IDO1. IDO1 is a fascinating protein and can be considered a moonlighting protein. That's why the title of my talk is Magic in the Moonlight. It is a title of a nice movie, but it also refers to the fact that IDO1 is a moonlighting protein. And for those of you who are not familiar with this concept, a moonlighting protein is a protein that has a main activity, but besides that, it has also some side activity. So in other words, it is a multitasking protein. And these proteins change from one task to the other just by changing conformation in response to different environmental conditions. To give you an idea of how many moonlighting proteins have been identified so far, here you can see a nice picture of one of our review paper on that, and it represents the moonlighting proteins that have been identified so far and have a role in the immune system. And among these proteins, you can see that there is also here, very small in purple, IDO1. IDO1 is a protein that has a catalytic activity, and you can see it on the left, but it has also signaling activities on the right. Why have we been working on IDO1 for the last few years? Because IDO1 plays an important role in immune tolerance. So under physiological condition is the protein, thanks to this protein, the fetus is not rejected by the immune system of the mother, for example, but under pathological condition, it is a protein involved in immune escape. So it contributes to the formation of an immunosuppressive microenvironment. And this is due to several mechanisms that I'm not going to explain now, but there is a direct correlation between the overexpression of IDO1 and the tumor progression and the poor prognosis in oncological patients. However, out of the thousands of IDO1 inhibitors that have been synthesized so far, no molecule has been approved by regulatory agencies, and it is still not clear whether the IDO1 inhibition could be an effective strategy in the immunotherapy of cancer. And in 2018, the most advanced IDO1 inhibitor at that time, epacadostat, failed to be effective when compared to pembrolizumab in a combination with pembrolizumab. And this was done in patients affected by melanoma. And several reasons have been given in order to explain this failure. But it is thought that the main reason for this failure is that at the time of the discovery of epacadostat, the knowledge about IDO1 biology was really poor. And nowadays it is clear that IDO1 is a much more complex protein than what was initially assumed. So I will focus my lecture on 3 different key features that IDO1 has. And I will also highlight how you can modulate these key features by using small molecules. So I will focus my lecture on the evolution that the understanding of IDO1 has experienced over time. So the first key feature is that IDO1 is an enzyme. So it catalyzes tryptophan into formylkynurenine, which is then hydrolyzed into kynurenine. It is a monomeric protein. It is induced by the cytokine interferon gamma. And this protein is composed of 2 different domains, a small domain and a large domain where the catalytic activity takes place. And here you can see the heme group, which is an essential cofactor for the catalytic activity of IDO1. So it is not surprising that most of the IDO1 inhibitors reported so far are metalloenzyme inhibitors that can bind to the heme and the iron of the heme through a nitrogen atom, as in navoximod, or through an oxygen atom, as in epacadostat. And usually, these compounds bind to 2 pockets, the pocket A, which is a small hydrophobic pocket here, and pocket B, which is an outward pocket at the entrance of the binding site. And let me draw your attention to this imidazothiazole compound, because it was reported by our group, and it was the first compound that was reported to extend into a 3rd pocket that we named pocket C, forming a key hydrogen bond with the lysine residue. The second feature of IDO1 is that if we consider a cellular environment, IDO1 is in a dynamic equilibrium between a holoform, which bears the heme group, and a hapoform, which is devoid of the heme group. And while the holoform is catalytically competent, the apoform is not catalytically competent. And the step of heme release is a very slow step and is a temperature-sensitive step. This information will be useful throughout my presentation. What it is interesting to say is that up to 80% of IDO1 can be found in the cell in its apoform. It has been demonstrated, for example, that in tumoral ovarian cells, IDO1 can be found almost in its apoform, up to 85%. So in 2018, the same year as the failure of epacadostat, the first APO inhibitors started to appear in the literature. And nowadays, the most advanced APO inhibitor is lirudostat. You can see the compound here. Lirudostat, and in general, these APO inhibitors bind to the apoform of IDO1 and prevent the reloading of the heme group. Lirudostat binds to pocket A and also pocket D in yellow, which is formed after the release of the heme, and it is quite large and hydrophobic pocket. Typically, APO inhibitors show a slow off rate and prolonged target engagement, so they seem to be superior inhibitors compared to the holo inhibitors. The third feature is that IDO1 has a signaling activity, and while the catalytic activity occurs in the large domain, the signaling activities occur in the small domain where ITINs are present. And in particular, after a conformational change of the proteins, ITINs are exposed and are phosphorylated in correspondence of tyrosine residue. And this promotes the association of IDO1 with different molecular partners. And in particular, in a context driven by interleukin-6, IDO1 is able to associate with SOX3, and this induces the proteasomal degradation of the protein, while in a context driven by TGF-beta, IDO1 associates with the sheets, and this induces the de novo synthesis of both IDO1 and TGF-beta itself. So we can state that IDO1 is a protein that has the ability to fine-tune its half-life and to fine-tune the equilibrium between immunity and tolerance by regulating its own expression. But there is a third thing that IDO1 can do. You can see here there is a Yen motif, and when this Yen motif is phosphorylated, IDO1 can also associate with PI3 kinases, and this causes a switch of IDO1 from the cytosol to the early endosomes. And it is important because the catalytic activity seems to take place in the cytosol, while the signaling activities seem to take place when IDO1 is anchored to the early endosomes. So in our lab, a few years ago, we did a structure-based virtual screening. And curiously, by a single structure-based virtual screening, we were able to identify 3 compounds with a different mechanism of action. So you can see here VS9, which is a holo inhibitor. Here you can see VS13, which is an APO inhibitor. It is a very potent compound with an IC50 of 16 nanomolar in a cell-based assay. But we were also able to identify this compound here, named VS15, which has an unprecedented mechanism of action. And that's why in the next slide, I will focus on this compound here, because it is able to negatively interfere with the signaling activities of IDO1 and not only with the catalytic activity of the protein. So the first thing is that VS15 is able to inhibit IDO1, and it is able to do that in a melanoma cell line where IDO1 is induced by the treatment with interferon gamma, but it is also able to inhibit IDO1 in a mastocytoma cell line transfected with both human enzyme and urine enzymes. So you can see that, you cannot see that, but the IC50 is identical. So this is quite important from a development point of view. And this inhibition is strictly related to the inhibition itself and not to cytotoxic activity, because the compound is not cytotoxic up to 30 micromolar and is not related to a degradation of IDO1. So the reduction that we see in cells of kinurane is not related nor to cytotoxic effect, nor to an interference with IDO1 degradation. In spite of the presence of this decarbonyl function, the VS15 is quite stable in different buffers. So all the data that I will present are quite reliable. The interesting thing is that when we perform a biochemical assay by using the human recombinant protein, we didn't see any activity at all. So this is epacadostat, this is VS15. And so we were quite intrigued by it. But when we perform the same experiment under forced conditions, in particular by increasing the pre-incubation times and by increasing the temperature, we could see an activity. And this is suggesting that VS15 is an APO inhibitor. So it is active under those conditions that favor the heme release. So we did other 3 experiments to confirm this APO inhibition. A cell-based experiment in the presence of acetone, which is an inhibitor of the heme biosynthesis. And you can see a shift of the dose response curve and a decrease in the IC50 value. We also did a spectrophotometric experiment where we evaluated the Sorey band. A Sorey band is an absorbance peak, which is typical of proteins bound to the heme group. And here you can see over time the disappearance of this band at 404 nanometers, which suggests that there is a displacement of the heme. And also in another biochemical assay, we could see an increase of the free heme group in the presence of VS15 compared to the control. So we tried to do an X-ray crystallography study, but we didn't succeed, probably because of solubility issues. So this is just a docking pose, but the binding mode seems to be very similar to other APO inhibitors, where VS15 stands from pocket A to pocket D, forming key hydrogen interactions. But the most interesting thing of VS15 is that it can negatively interfere with the signaling activities of IDO1, and in particular, it can decrease the association of IDO1 with PI3 kinases, and this leads to the fact that the protein is retained in the early endosomes compared to the cytosol. It can also inhibit the association with the phosphatases SHIPs, and here I thank Esther for the lecture that was given before on these phosphatase enzymes, and as you can see, the interaction with SHIP1 and SHIP2 was reduced, and this causes also the reduction of the formation of free phosphate in a phosphatase assay. And as a result, VS15 is able to decrease the expression of both IDO1 and TGF-beta. So based on these results, we wanted to investigate whether YES15 was able to reverse the immunosuppressive phenotype. And so we co-cultured T cells together with the monocytes, suppressive monocytes expressing IDO1 coming from patients affected by pancreatic cancer. And we could see that in the presence of YES15, the reduction of proliferation of T cells in the presence of these immunosuppressive monocytes was reduced, significantly reduced. So in summary, we have a compound which is an APO inhibitor, which interferes with the signaling of activities of IDO, but unfortunately, this compound is not metabolically stable. So it's chemically stable, but due to the hydrolysis that occurs on the oxalilamide and to several oxidation reactions in other sites, is not metabolically stable. So we started a medicinal chemistry campaign aimed at discovering compounds that retain the same potency while having a good metabolic stability. We changed the 4 portions of the molecule. We synthesized 26 derivatives. And the most interesting compound so far is compound 11, which has quite a good metabolic stability compared to YES15 and also retains the same potency. So we wanted to verify that this compound is still an APO inhibitor and still interferes with the association of IDO1 with sheets, and we verified that. We also did a metabolomic study by using liquid chromatography with a tandem mass spectrometry method developed by our collaborators, and we could see that this compound is also able to reduce the level of the downstream metabolites of the kynurenine pathway. So in this slide, the different modalities that we explored for targeting IDO1 are summarized. So we were able to reduce the catalytic activity of the protein, to reduce the signaling activities of the protein. And the lesson learned from this project is this one, that the easiest way of screening compounds is not always the best way to make a discovery. Why I'm saying that? Because at the time when we initiated this virtual structure-based virtual screening, we were not aware of the possibility that an APO inhibition could occur because it was not known in the literature. And we decided to perform the screening using a cell-based assay and not a biochemical assay in order to discard all those non-cell permeable compounds. And this was a lucky choice because it allowed us to discover APO inhibitors. So if we had used a biochemical assay, we would have never discovered YES13 and YES15. There is another last modality that I would like to show to you. Obviously, in order to get rid of all the activities associated with IDO1, the best way is to degrade the protein. So as other medicinal chemists have done in this last period, we went for the PROTACs. So I think that there is no need for explaining how a PROTAC works, but I would like to show to you a platform, a synthetic platform that we conceived in our lab. This platform allows the synthesis, the straightforward synthesis of PROTACs in just one step by relying on a type of chemistry that we really like in our laboratory. This is multi-component reactions, and so this platform is based on the use of the Ugi reaction, the Passerini reaction, 2 variants of the Ugi reaction that we discovered in our lab. And so in just one step, by using a quite reliable and versatile platform, we are able to synthesize PROTACs quite easily. And so we obviously apply this platform also in the synthesis of PROTACs. So we were able to easily vary the length and the nature of the linker, the nature of the anchor core for the E3 ligase, also the nature of the attachment points. And we used as a warhead an analog of the YES13, that APO inhibitor that I told you is very potent. So these compounds are now being evaluated for their inhibitory and degrading activity. It is quite a new project, so I don't have any biological data to show to you, unfortunately. And to conclude, let me thank my team, which is made of young and talented researchers. You can see that we span from 1.5 meters to 2 meters. And we also have Yannick Steiner here. We call him Yannick because he really looks like the tennis player. And yeah, I also thank the collaborators that work in my university, at the University of Perugia, at the University of Verona, you for your kind attention, and I'm open to any questions.
Tracey, see we have a few questions. So.
Very nice. Well, thank you. So you said that we wouldn't have discovered it if it wasn't for the study you're asking, but computationally, was there a way that we could discover it? I mean, was the pocket there, or the pocket was in the conformation of the assembly that we should not be able to discover? Maybe with cryptic pocket identification, but
yeah, what is curious about this visual screening that we perform is that it was made on the chloroform of the enzyme, but in spite of that, we were able to identify also APO inhibitors. Now we are doing, but this is another story, we are also performing, we have performed a visual screening using the APO form of the protein, and we have discovered other APO inhibitors.
But was the pocket there or not?
In the first visual screening that we performed, no, because we used the follow structure of the protein, so there was not the pocket D, that is the pocket that usually binds to, APO inhibitors bind to.
But in the
So the
APO
pocket
form,
was
it is there.
occupied by the heme group.
But in the APO form, it is there.
Yeah, in the APO form, the heme group is not there. So we performed the first visual screening in the presence of the heme group. But in spite of that, we were able to identify APO inhibitors. But probably the reason is that it has been reported that sometimes these APO inhibitors bind to the hollow form of the protein and then causes the displacement of the heme group through a suicide mechanism of action. We don't know how the S15 behaves, but there is a possibility that it does not bind to the APO form directly, but it first binds to the hollow form and then causes the displacement of the heme group in a second moment.
Thank you.
Welcome.
Was another question? Yes, please.
Thank you for the very interesting talk. So honestly, I was not very familiar with the moonlight proteins. So my question is, when we think about all these proteins identified as moonlight proteins, so what are the key aspects that they have that gives them the multitasking feature? And when we compare it with the IDO1, what can you say about the activity and the structures?
Well, I think that all these moonlighting proteins share a peculiarity that is the conformational plasticity. So usually the common feature of these proteins is that they can change conformation according to the environment, according to the different cytokines, for example, if we talk about the immunological system. And indeed, the conformational plasticity is another feature that I didn't have the time to mention in my lecture, but characterizes IDO1 as well. And so, yeah, usually they have a catalytic activity, but besides from that, they also partner with other proteins and also participate in other signaling pathways. This is the main feature of these moonlighting proteins. This is mainly discovered by serendipity, I mean, maybe doing by some co-immunoprecipitation experiments where you can see the association, and then you start thinking that you have a moonlighting protein in front of you. There is not a rational way to find out moonlighting protein. It's just by serendipity.
Right, very well. There are no more questions. Then thank you again.